So protein synthesis, it requires a lot of energy. Let me see, is there a question in the chat? So it forms into a protein kiln, the correct protein. Once it detaches, it's gonna fold into a protein. So it requires a lot of energy, protein synthesis. So when you eat food, the food goes in your body and your body starts to make proteins, but your body is not making proteins all the time. It only makes the proteins that are needed. So like I said earlier, maybe it's gonna make a protein for the muscle fiber. Maybe it's gonna make protein for insulin. It's only when it's needed because it takes a lot of energy. It doesn't do it all the time. And we regulate the protein synthesis in different ways. This picture here is just to show you, this is death cap mushroom, and it can basically block the protein synthesis, and that's how it causes death. So this can block it completely. Let's talk about mutations next. So a mutation is when that sequence changes. So maybe there was supposed to be A there, now it's a G there, and now you have a different information on mRNA as well. So you have a wrong sequence. So this mutation example is there's one gene. When there's a mutation in that gene, it starts to develop heads where the antenna is supposed to be. So it starts to develop legs on the head, basically. So a mutation is a change in a DNA sequence, and they can be in several varieties. There can be a mutation that's only changing a single letter. There can be a mutation that's basically changing a whole codon, so 3 letters, or it's deleting a codon, or it's inserting another different codon. So there can be different ones. So let's look at that. So I'm going to use English language for example. So a point mutation, it's a small mutation. It's a few base pairs in a gene. So few base pairs change in a gene. So this is your original sequence. The one big fly had one red eye. That's your wild type. That's your original. There is something called substitution mutation. Now one letter changed here. This doesn't make sense. This still makes sense, but this doesn't make sense. One letter got substituted for another one. Now, if these 3 letters stand for the same amino acid, then there's no change. But if this stands for a different amino acid, then it might be a different protein. This one is called deletion of 3 nucleotides, so deletion of a codon. So this is your original. The one big fly had one red eye. Now you have one big had one red eye. This doesn't make sense. The codon got deleted here. And then there's insertion. The one big wet fly had one red eye. So there's insertion of 3 letters, which means a codon. So that's insertion. The very, very dangerous one is this one. It's called frame shift mutation. This one here, this is, again, let's read the wild type again. The one big fly had one red eye. So you still had the one, but then a single letter got inserted and it just shifted the whole sequence. It shifted everything. Now this is just gibberish. It makes no sense. So this is a very dangerous, lethal type of mutation. And then again, remember that codons are sequences of 3 nucleotides. So 3 nucleotides make a codon. So these are definitions now. So substitution, mutation, changing one letter. So that's the wild type. So you have nucleotides that changed. And then insertion, in this case, deletion would be deleting a codon. Insertion would be inserting a codon here. So that's insertion. And then the frame shift mutations affect multiple codons because it just shifted the whole nucleotide sequence. So every codon is affected after this, once that letter got inserted. The type of question on the exam that you can expect, I will give you a sequence, and then I'll say this is the original sequence. Then I'll give you another sequence with maybe there's 3 letters that are inserted or 3 letters that are deleted, or this, it looks like this. Maybe I inserted one and then whole frame changed. So you're gonna have to tell me if it's a frame shift, if it's an insertion, if it's a deletion, if it's a substitution. So try to kind of go back and look at it one more time. Are there any questions on mutations? No? So insertion or deletions of 3 nucleotides add or delete entire codons. So, but they do not affect other codons. You can still read the other codons, whereas here you can't read any of the codons after this. So let's talk about sickle cell anemia. You guys heard about sickle cell anemia disease? So the normal shape of your red blood cells is like this. It's a biconcave shape, biconcave disc. And the reason it has this shape is so it doesn't clog the blood vessels and flows in and out easily. This is the DNA sequence for it. And this is, transcription into mRNA. Then these are the amino acids. If there is a mutation, so let me show you something. So this is what your normal blood cells look like. But if you have that mutation, then the blood cells take this shape. This is called sickle cell anemia, because this looks like sickle knife. So that's the shape it has. So here, so that's the shape it gets. So that's where the name comes from, sickle cell anemia. So this is what they start looking like. And then they start to clog up the arteries or the vein or the blood vessels. So here you can see they are starting to clog it up. And the person who has severe sickle cell anemia, they're in pain all the time. So here is a mutation, only one letter changed. Now, what type of mutation is this? So what type of mutation is this? Substitution, yes. You can even see that here, single base substitution. So there's a substitution mutation, one letter changed. But what happened, these 3 letters stood for pro, GAG stood for glue, and then GAA glue again. Because when this one letter changed, now you have still pro, but this changed to valine, VAL, and then you still have glue, and that changed the shape. Now, if it's just one gene and it's not very severe, it's mild type of sickle cell anemia, then the person is still okay. But in the countries where they have malaria, mosquitoes, they can basically affect this shape, but they're not able to penetrate this shape. So the people who have mild sickle cell anemia, they don't get malaria because of this shape. So you're gonna see wherever there is high incidence of malaria, it's gonna be, there are gonna be people who have sickle cell anemia. And then in the United States, people who have African heritage have higher incidence of sickle cell anemia. So you can see here versus other people. So I guess that's where the mutation started. So mutations, they're not always harmful. There can be mutations that are beneficial. And then humans these days, us, we start to, use biotechnology a lot to maybe change things to our benefit. So maybe, some tomatoes that you get in the market, they're really big in size. That's because we maybe changed some of the genes and made a hybrid. So mutations, they're not always harmful. So another definition I really need you guys to know, because this is gonna come up again and you're gonna get confused. So it's called allele. So mutations, they create different versions of the gene, of the same gene. And that's called allele. So in this picture, The citrus has this reddish color flesh. Now this can be yellow. That's the same gene, just a different version. That's an allele. So I need you to know the definition of allele. Now, if you think about nature, this lemon, it's, we made that lemon by changing some of the stuff. So lemon tastes sour, and the whole purpose of the fruit for plants is to entice animals so they can basically disperse the seeds. So which animal would like to eat the sour lemon? So that's something that we made from the citrus. So genetic variation is important for evolution. If there's a mutation that's beneficial, it's gonna stick around. Think about giraffes. They have really long necks. They can reach, for food at very high, at higher places, so very high trees or something like that. If they didn't have that mutation for a long neck, they wouldn't be able to reach for food. Maybe they will die out. So that's just an example I'm trying to give for evolution. So if a mutation is beneficial, it's gonna increase in population. It's gonna stick around. Person who doesn't have that mutation that's beneficial, maybe for them for living, then they might die out and they won't be able to pass on their genes. Plant breeders, they induce mutations to create new varieties of plants. Sometimes maybe a plant has a gene that prevents maybe ants can't eat the plant now. Maybe there's a mutation and it makes some type of toxin that would kill the ants. So if we like that, we might be able to take the gene and insert it into a plant that gets eaten by the ants. So that plant becomes resistant to the ants if we do that. So plant breeders do that. They induce mutations to create new varieties of plants. So there is a question for you guys. The DNA template strand sequence below mutates. It's showing a mutation. What happens to the amino acid encoded by these two codons? And your answer choices are both amino acids change, only one amino acid changes, and neither amino acid changes. So what I need you to do right now, I want you to write down this original sequence, and then I want you to write down this mutated sequence. So I'm going to go to the chart next, and you're gonna tell me which one is true. So go ahead, I'm gonna give you a minute to write this down. Write down the original sequence and then the mutated sequence. So I'm gonna go to the chart now. Another thing I need you to keep in mind when you're reading the question, if it's a DNA sequence that's given to you or it's the RNA sequence that's given to you. So going back here, it says a DNA template strand. Once you are done, go ahead and type your answer in the chat. So choice A was both amino acid change, B, one amino acid changes, C, none of them change. Okay, so what I said earlier, I said when you're reading this type of question, there will be 3 questions on the exam worth at least 3 to 5 points. When you're reading this type of question, you want to keep in mind if it's a DNA template or RNA template. Because if it's a DNA template, then you need to convert it into RNA first and then the protein. If it's RNA already, then you can just go to the chart and get the protein. So let's do this one together. So I'm gonna say this is our DNA original sequence for the original. We have A, G, T, and then we have T, C, T. So I'm working on this original sequence right now. So this one, the one I'm highlighting. So for that, we need to convert into RNA first. What would you have the RNA complementary for AGT? What is the RNA complementary for AGT?
U,
U,
C,
C, A,
A. And what about TCT?
A,
A, G,
G,
A.
A.
Now we do the protein. So we go on the chart, we look for UCA. First position, U. Second position, C. UCA stands for SER. So that's your first protein, SER. Then you have AGA. That's your second protein. First position, A. Second position, G. So you're gonna look in this box here, A and G, and AGA stands for ARG. So that's your second amino acid. Now let's work on the mutated sequence. So for DNA sequence, you have GGT and TCC. For RNA, what are you gonna have for GGT?
CC,
CC,
A,
A, and for TCC,
A,
A,
GG.
GG. So for protein, now you're gonna go to the chart, CCA. So you have first C, so you're looking in this row, and the second letter C, so you're looking in this box. CCA stands for PRO. So that's your first protein. Second one, you have AGG. So let's look for that. So you have A, looking in this row, G, so down here, AGG stands for ARG. So that's your second protein. Now, what is the answer? What do you notice? How many proteins changed from here to here? Or how many amino acids changed, not proteins? One, right? You still have ARG here, only this one changed. So B would be the answer. Before I move on, I really want to make sure that everybody understands how to go from DNA to RNA and then RNA to protein. And in addition to that, everybody needs to know how to read the chart.